Hello everyone. So I welcome you all to the class of genetics. Yes, in this NEET Power Punch batch, we are doing genetics, and today is the day where we are going to solve some questions from the topic which is monohybrid cross and dihybrid cross. I hope that I am live and I am clearly audible and clearly visible to each and every one of you. Hi Sid, how are you? That's good to see you here. Okay, so now in today's session, sorry, our most focus will be to solve the questions of monohybrid and the dihybrid cross. But we have some concepts left like test cross, reciprocal cross and all these things. So these concepts we will be going to do in today's session only, so that we can clear the this Medelian genetics today only. From the Monday we will start with the post Medelian genetics. So starting with these question which we have done yesterday, if you have any kinds of doubt in this question, go to the last class video, watch that lecture, and not now, after this class, and clear your doubts. Okay, and in this case, if today if you're having any kinds of doubt, what you have to do, you have to just comment down there. I'll repeat that particular part. Okay. Let's revise things quickly. We have discussed about the monohybrid cross. Sir, what was monohybrid cross? Monohybrid cross was the cross performed between the two parents, and in that we were studying the inheritance of a single character. In that we saw, sir, that in F1 generation we have a plant which was all tall. Yes or no? That's very good. And what was the genotype of that particular plant, everyone? Yes, what was the genotype? The genotype was capital T and small t. This was all about the F1 generation, right? Okay, let me know if I'm going to write some things wrong, because I am going to do, and I'll check that whether you are paying the attention or not. Now, let's have the F2 generation. So what we did in F2 generation, guys, we self-crossed, right? What we did, we self-crossed. So selfing means what? We are going to use the progeny of F1 generation and we are going to cross it, right? So this is what we did. And what was the phenotypic ratio? Was it 4:1? Was it 4:1? No, sir. It was 3:1. It was 3:1. That's very good. Now, if I am going to talk about the 3:1, so in this 3 plants were tall and 1 plant were short. It was tall and short. See, I am revising all of the things so that if you are watching this video for the very first time, you won't have any confusions and you will be able to solve the questions. That's our main motive. And if you revise n number of times, that is going to enter into your brain and you will never forget. That's the point why I am making revisions in each sessions before starting the concept. Now let's say what this was all about, the monohybrid cross. And here sorry, what was the genotypic ratio? So the genotypic ratio was what? It was 1:2:1, sir. One plant was homozygous tall, the other 2 plants were heterozygous tall, and the one plant was what? It was homozygous tall. Is it clear to everyone? If it is yes, let me know in the comment section. That's very good. Now let us start with some of the things. What things, sir? We are going to have some of the questions that we are going to solve now. Okay, that's great. Okay, before that, should we revise the dihybrid cross also? Yes, definitely. So what was dihybrid cross? If I'm talking about the definition, so how can we define the dihybrid cross? We can define the dihybrid cross as a cross which is performed to study the inheritance of the characters. How many characters? 2 characters simultaneously, right? Now, what was the ratio F1? In F1, sir, we saw that all the plants were having the dominant character. What was that, sir? Yellow and round. Yes, that's correct. Now, in F2 generation, what did we had? We had the phenotypic ratio as 9:3:3:1, and this was the phenotypic ratio in F2 generation of dihybrid cross, and 3:1 was the phenotypic ratio in monohybrid cross. This much is clear, sir. Now we are ready to do the questions. Yes or no? That's very good. So let's quickly start with the questions, right? So I have a beautiful questions with you. We are going to solve the PYQs also, just from the dihybrid and the monohybrid cross, okay? But before that, let's prepare ourselves so that we are able to solve those PYQs questions, okay? Great. So now let's suppose that I'm giving you something that in a Mendelian cross, about 1600 plants, okay? How many plants? Let's suppose that in a Mendelian cross, 1600 plants were produced. Clear? So how many plants produced, sir? 1600 plants are produced. That's very good. Next, what? Next we have that this was what? This was the monohybrid cross. In a Mendelian monohybrid cross, the question says that in a Mendelian monohybrid cross, 1600 plants were produced. Okay, great, sir. Now I am going to ask in F2 generation, how many plants were dominant? How many plants were dominant? How many plants were dominant? Can you say this? Can you see this? That's a very basic question. I'm going to give you around 30 seconds to think about it and solve it. It's really a question that can be solved within 30 seconds. Okay? Okay. Solve it right now, right away. Should I give you a hint or something like that? Do you want me to give the hint? No, never believe me giving you the hints. I can give you the wrong hints also. Okay, now it's fine. Let's solve this question. Sir, we saw that first of all how you have to approach this question. See, how many progeny? 1600. Okay, how many plants? It has been produced. 1600 plants is produced. Okay, among the 1600 plants in the F2 generation. So now if I talk about the F2 generation, first of all, you remember the phenotypic ratio. It's asking about the phenotypic ratio, right? Like which is dominant, which is recessive. Okay, how many plants are dominant, how many plants are recessive. So what we have to do, first of all, remember that phenotypic ratio. So it was 3:1. What 3? 3 were dominant and 1 was recessive. So in this question, it has been asked that how many plants were dominant. So in total, what we are going to do is that we are going to divide 3/4. This was the ratio thing. Why, sir? Why we did 3/4? See, let's talk about this thing. Okay, let's talk about this thing. So in this thing, let us assume that what was the total number of plant? What was the total number of plants? So total number of plant were 4. Let's suppose in this case, total number of plant were 4. And among 4 plants, 3 of them were what? 3 of them were dominant. Okay. So the ratio will be what? 3/4. That means this is the ratio for the tall plant or dominant plant. So similarly, here to calculate the number of plants which is dominant, we have to multiply it by the ratio. So what we are going to do, 1600 multiplied by 3/4. And now I think so you know this basic mathematics, you can solve it. 4 1s are 4, 4 2s are 8, 4 3s are 12, 4 4s are 16, 0 0, 4 3s are 12, 0 0. So the answer is 1200. Yes or no? See this? Clear to everyone? So 1200 plants are what? They are dominant. Very good. That's very good. We did the first question. Now, should I move forward? Definitely, yes, you don't have any other options. Okay, now I'm going to give you another question. Should I give a little hard question? Yes. Let's solve a question from a dihybrid cross. Okay. So let's suppose a genotype heterozygous genotype. What heterozygous genotype means? Heterozygous genotype means that the genotype is having two different alleles. Okay, heterozygous genotype for capital Y and capital R forms what? Forms how many? One second. Forms how many gametes? So what I am saying that there is a heterozygous genotype for Y, capital Y and capital R, and the question is asking that how many gametes they are going to form. Okay, forms how many gametes? It's a very easy peasy question. It's a very easy peasy question. Yes or no? Yes, sir, it is. Then solve it quickly. What we are going to do here? First of all, the question didn't give you the exact genotype, but the question maker said that it is heterozygous, and he has given you Y and R. That means what? That means firstly you have to find out the genotype. Okay, so we have find out the genotype. What is genotype here? Heterozygous genotype for both Y and R. So if we have one capital Y, one which is dominant, so the other one will be what? The other one will be the small y. Yes or no? And if we have the capital R here, so the other one will be what? Smaller. See, we have got the genotype. We have got the genotype, and as soon as we have got the genotype, they have asked how many gametes. How many gametes? Do the fork line method. That's very good. What do we need to do? We need to do the fork line method. Clear? Okay, great then. Great. So how we are going to do the fork line method here? See, we have studied that. Yes or no? If you have forgotten, then I can't do anything. You have to revise. Remember that you are going to give NEET on 5th May, and if you are able to forget things, then your revision is not working good. Okay, so now bifurcate it. This is the one heterozygous pair, this is the another heterozygous pair. What we have to do? We have to bifurcate. So this is capital Y and this is small y. Done, sir. Done. Okay, first step of the gamete formation is done. Capital Y and small y, right? Now what? Now we are going to bifurcate for R. Because why? Because it is also what? It is also the heterozygous condition. So capital R and small r. Here also we will be having capital R and small r. Done? Yes, sir. Done. Now what? Now we need to figure it out. Now we need to figure it out that how many types of gametes we have formed. Okay, I'll be little here so that you can see the screen properly. So this is Y, this is what? This is Y, and this is what? This is capital R. So here we are going to have capital Y and capital R as one gamete. Okay, and here we have what? See, here we have capital Y and small r. So what type of gamete we are going to have? We are going to have capital Y and small r. Yes or no? In a similar manner, we have what? We have small y here, and if we have small y, and here we have capital R. So what kind of gametes we are going to have? We are going to have the gametes of small y and capital R. That's clear. Now here we have capital Y and small r. Capital Y? Small y, sir. What is this behavior? Sorry. So we have small y, small r. Yes or no? How many types of gametes, guys? We have, sir, 4 types of gametes. Now, what will be the percentage of each of the gametes? If you are good in maths, you can answer this question very quickly, but if you're not good in maths like me, we will take some time, okay? But don't worry, I know maths little bit so that I can teach you genetics. Now, let's move on. Let's move on with this thing, which was this. Okay. So in total, how many things we have? If suppose all the gametes will make up 100%. Okay. All the gametes will make up 100%. How we have total number of 4 gametes. So just divide it 100 divided by 4. Yeah. So it will be what? It will be 25. So each gametes are contributing 25%. Yes or no? Yes, sir. That's very good. Now, if you have any kinds of doubt in this particular question, let me know in the comment section. Let me know in the comment section. Clear? Okay. That's very good. Okay, said. No doubts. We are going to move forward then. Now, let's have, okay. If I ask you about the ratio thing, so how you are going to write the ratios here? Like you are going to write 1:1:1:1. Okay, great, clear, that's good. Now, let's move on and let's have the another question with us. Okay, great. So now I am going to give, okay, give me a hint. Should I give you the question with the monohybrid cross or the dihybrid cross? Which question do you want? Fast. Tell me fast. Okay, I'll give you the question from dihybrid cross. Okay, take it. Next. So the question says that in a Mendelian dihybrid cross, in the Mendelian dihybrid cross, let me think about some numbers. In a Mendelian dihybrid cross, let's take 512. 512 plants, 512 plants were obtained. How many plants? 512 plants were obtained in F2 generation. Okay. Now, how many of them shows the dominant characters for both, dominant phenotype for both the characters? Okay. How many of them shows dominant character for, I should write dominant phenotype for both characters. Okay, phenotype for both characters. Clear? Okay, sir. I think so this question is very easy and you have to do this question because this is very easy. Yes. So, how we are going to solve the question, sir? We know that here that we have 512 plants. Okay, great. How many plants, sir? We have 512 plants. And first of all, in these type of questions, your phenotypic ratio should be in your mind. In monohybrid cross, it is 3:1. In dihybrid cross, it is 9:3:3:1. Okay, then write it down. 9:3:3:1 is the in the dominant in the dihybrid cross. Great. Now, this was the ratio. This 9 was the ratio where we can find what? Where we can find that all the characters in which this plant is having, like the yellow and all those things. So all the characters are showing it their dominant phenotype, and that was what? That was 9. So in 9, all of them were dominant. Okay, great, sir. Now, do you remember this question? We did it 3/4 into 1600. Yes or no? So similarly, how we are going to solve this question here? We are going to solve this question in a similar manner. How many total number of plants do we have in Mendelian dihybrid cross? Sir, 16. How 16, sir? How can you say this? I can say this by calculating here. 9 plus 3 is 12. 12 plus 3 is 15. 15 plus 1 is 16. Yes. So total number of plants is 16. Okay, sir, written. Now, how many plants are having the dominant character here? 9, sir. Great. Now what you do is that multiply it by 512. So easy. So easy. Now, when you do the multiplication here, what you're going to get? Do it. Waiting for me to calculate? Do it fast. And let me know the answers in the comment section. Fast. Fast, fast, fast. I'm seeing the comment section. I know you can give the answers. Fast. Do I have the answer here? Do I have the answers here? Anyone in the comment section? Oh, that's very good. We have an answer. 288. Okay, 288. 16 and 512. 16 and 512. So 16 ones are 16. And can we cut it directly? 16 ones are 16. Yes, this will be 32. And 32 into 9. 9 twos are 18. 9 threes are 27, 28. Yes, very good. So we have the answer as 288. Okay. Yes, Sid, what you can do is that after this class, buy a chocolate for yourself and celebrate it. Clear? Good job. Now, let's move on and let us talk about some other things. Okay. To solve the questions in genetics, it requires some tricks and tips. It requires some important probabilities. It requires some presence of mind. So all these things I'm going to discuss with you right away. Give your heading as some important probabilities. All the probabilities which can be used in solving the questions. All of those things I will be teaching you here. So write it down. Some important probabilities. Some important probabilities. Okay, great. Now let's see here. First of all, we need that probability of a homozygous genotype in F2 generation. If suppose that if suppose someone asked you that what is the probability of homozygous genotype in F2 generation of a monohybrid cross. Okay, homozygous, anyone can answer if you know. Homozygous genotype in what? In F2 generation in monohybrid cross. In monohybrid cross. Okay. How, sir? How can you find this? You have not taught us. I have taught you, but you are not able to remember, right? See? Okay. Now see. Don't worry. It's a very easy thing. When you understand this type of concept, there will be no questions which you are not able to solve. Every question will be a cakewalk for you. Now see. How many? Again, I'll ask the same question. What was the ratio for the phenotype in F2 generation of a monohybrid cross? Sir, it was 3:1. Total number of plants according to the monohybrid cross of Mendel. Sir, 3 plus 1 will be 4. Okay, let's write 4 here. And how many plants were homozygous? How many plants were homozygous, having the homozygous genotype? How many plants were having homozygous genotype? Think about it. Will it be 1 by 4, 2 by 4, 3 by 4 or 4 by 4? How many plants are having homozygous genotype? Sir, having confusions. Okay, great. So let's see here. If we have capital T, okay, we have what? We have capital T, small t, and then we have small t and small t. Okay, this was the genotypic ratio. Okay, this was the genotypic ratio. Clear? Okay, sir. This was the phenotypic ratio. Clear? Okay, sir. Now, can you see this one is also homozygous and this one is also homozygous? Yes or no? This one is this. Where should I stand? I will stand here. This capital T is homozygous and this small t is also homozygous. Okay, how many total number of plants did we had? 4. Then among them, how many plants are homozygous? So sir, 2 of them are homozygous. Why? Because this ratio says 1:2:1. So in total 2 of them are homozygous. So our ratio will be what? It will be 1/2. Is it clear to everyone? Is it clear to everyone? Clear? Now, in this question, I have asked you about the homozygous phenotype. Okay. Now, I can ask you the homozygous dominant phenotype. Then genotype, it will be what? It will be 1/4. Homozygous recessive phenotype or genotype, it will be 1/4. Clear? Okay, so that's great. Now, let us move on with the second thing. Second question, I'll be asking you that probability of heterozygous genotype in F2 generation. Next question is probability of heterozygous genotype in F2 generation of Mendelian cross. Okay, of Mendelian monohybrid cross. Same concept. Apply it. Same concept. Try to apply it. Try to calculate it by yourself. Try to apply it. Try to calculate it by yourself. Okay. Done? Lots of time I have given. Now let's solve it. So here, sir, here we will be having what? We will be having again 3:1 phenotypic ratio, 1:2:1 genotypic ratio. Total number of plants we have 4. Okay. And the ratios were what? 3:1 were phenotypic ratio and 1:2:1 was genotypic ratio. Okay. So in genotypic ratio, if I talk about this thing, which is this one, so this is the plant which is having what? This is the part which is having heterozygous genotype, and this is what? Among 4, 2 plants are like this. So this we are going to do like this. So again we have got 1/2 as our ratio. Remember that if they are asking homozygous dominant, if they are asking homozygous dominant, so it will not be 1/2, rather it will be 1/4, because homozygous dominant is only capital T and capital T. And if I'm talking about that homozygous recessive, then it will be also what? It will be 1/4. And if the question has not asked a single thing, not asked about whether it is dominant or a recessive, what do you need to do is that just write 1/2. Okay, clear? That's very good, sir. Now let us move on and let us talk about the number of phenotype in F2 generation. Okay, how we can calculate this, sir? See, if suppose someone asks you that this is the particular set of genotype, and let me know that how many phenotypes can be produced. Okay, so to calculate the number of phenotypes. So first let's go with the monohybrid. Let's talk about the monohybrid one. And second, let's talk about the dihybrid. Okay. Second, let's talk about the dihybrid. First is what? Monohybrid, and second is dihybrid. Clear? Okay, great, sir. So if I talk about this monohybrid thing, so in monohybrid we have a formula 2:n. What we have a formula? We have a formula that is 2:n. In a similar manner, for dihybrid cross also we have a formula 2:n. Sir, what is n? The question here is, what is n? N for nest? No. N is not equal to nest here. N is what? N is basically number of what? Number of, or I should say the types of the phenotype. What? N is the type, N is the thing which is going to give us the types of the phenotype. So sir, what is N? N is the number of heterozygous pairs. N is what? Number of heterozygous pairs. So we are able to find N, that N is what? Number of heterozygous pairs. What is N, sir? N is the number of heterozygous pairs. Clear? Okay, great. Now, if I give you the question, suppose that if I give you the question, find the phenotype, or I should say find the number of phenotype produced by capital T, small t. Okay, how you're going to do? See, we know that how many types of phenotype will be produced in this question, but if something else comes which you don't know, then that will create an issue for you. So number of phenotype, to calculate the number of phenotype, what we need to do is that we need to do this formula. We need to apply this formula which is above my head right now, 2 to power n. What is the formula? The formula is 2 to the power n. Clear? Okay, great. Now what? Now in this, how many types, like how many heterozygous pairs do we have? We have one heterozygous pair. So 2 to the power 1 is what? 2 to the power 1 is what? 1? Is it so? 2 to the power 1. 2 to the power 1 is equal to 2. 2 to the power 1 is equal to 2. So how many types of how many types of phenotypes will be produced? 2 types. Obviously, one is tall, the other one is short, with proof, right? With proof that this formula works. We read that in plant height we have tall and short plants. See? So how many phenotypes? 2 phenotypes. One will be tall, the other will be short. One will be tall, the other will be short. Great. Now, again, the same question with different genotype. Let's practice more. The same question with a different genotype. The same question with a different genotype. See. The genotype, okay, I'll change the genotype. Don't worry, give me some time. The genotype thing, let me think. Okay. Let me have the genotype as capital Y, small y, capital R, small r. Okay. Capital Y, small y, capital R, small r. Okay. How many pairs of heterozygous alleles we have? N represents what? N represents the number of heterozygous pairs. So this is what? Capital Y, small y, 1 pair. Capital R, small r, 1 pair. And if you don't know how to see the heterozygous pairs, you need to watch the video for the basic terminologies of genetics, the first class of this NEET Power Punch series. And tomorrow we will be having a series in which we will be solving lots and lots of questions from cell cycle, right? Okay. Now, here what's the value for n? The value for n is nothing but the 2. The value for n is nothing but the 2. So 2 to the power 2 will be what? 4. So yes, 4 types of phenotype is produced. How's that? Give us the confirmation. Okay, see, did you remember we studied 9:3:3:1? Yes. So in this, how many types of phenotype is produced? 1, 2, 3, 4. Don't say 16. 16 is not the phenotype, it's the number of progeny. What is this behavior, guys? 16 is the number of progeny. 4 is the number of phenotype. Clear? Okay, now let's move on. Why I have erased? Okay, fine. Happy now? You are able to solve the question if someone asks you that what is the number of phenotype or how many phenotypes are produced. Clear? Okay. Similarly, I will give you something for the genotype as well. I'll give you something for the genotype as well. Okay. So let me write here genotype. Sir, how can we do this? So, sorry. The formula for the genotype is what? The formula for the genotype is 3 is to power n. Formula for the genotype is 3 to the power n, while to find the phenotype it is 2 to the power n. Clear? Okay, great. How can we remember, sir? We will get confused. We are a small kids, right? We will get confused whether 2 to the power n was phenotype or 3 to the power n was phenotype. So, see, you see something from the outside, right? So 2 comes first, then you will go and enter its cell, then you're going to see the genes, then 3 comes. Okay, so 2 for phenotype, 3 for genotype. Clear? Okay, formula is same, n is same. Here also n is what? N is the number of heterozygous pair. Do I need to write again? Yes, sir. Number of heterozygous pair. Why, sir? Because you're gonna forget it. Like, what was n? What was n, right? So n is number of heterozygous pairs. N is the number of heterozygous pairs. Clear? Okay, let's do the questions. Capital T, small t, capital Y, small y, capital R, small r. Okay, let's do this question. Yes, sir, let's do this. So how we are going to solve this? We are going to solve by the help of the formula, which is 3 to power n. What's the value for n here? How many heterozygous pairs can you see? 1. Yes, 3 to the power 1. Clear? In this case, how many heterozygous pairs we can see? In this case, how many heterozygous pairs we can see? This is capital Y, small y, capital R, small r. 2. So 3 to the power 2 will be what? It will be 3 3s are. What will be it? 3 3s are 9. Exactly. 3 3s are 9. And here we have 3 1s are 3. So in total 3 genotypes are produced in this thing, and here 9 genotypes will be produced. Clear? Okay, got it, sir. Now what? Now we have number of zygotes. Now we have number of zygotes. How many types of zygotes, how many kinds of zygotes can be formed? So here is also a very good trick. So just remember, there is a plant, from outside you saw, and let's give it the least number. Outside you saw the phenotype is 2 to the power n. Then you entered the cell, inside the cell, then give it a number as 3. That means genotype is 3. And then these cells produce the babies. That means zygote will be 4. Is it clear? So for what? To find the phenotype, you give the least number 2. To find the genotype, plus 1. To form the zygote, plus 1. So how many we are going to have? We are going to have 4 to the power. We are going to have 4 to the power n. How many, sir? 4 to the power n. Clear? N is what? Again you are asking. N is the same thing. Heterozygous. Number of heterozygous pair. N is what? N is the number of heterozygous pairs. Heterozygous pairs. Yes, I'll move here from. See, now is it visible? Clear? N is what? Number of, which hand should I put? This one? Yeah. N is the number of heterozygous pairs. Again, let's do the practice here. So I'll going to give you the question, the same thing. Let's take capital T, small t, and let's take capital Y, small y, capital R, small r. Clear? Okay, sir. That's very good. Now what? Now we are going to calculate the number of zygotes. So 4 to the power n, which will be 4, and here 4 to the power. How many types of heterozygous pairs do we have here? We have 2, and can you get it 16 here? Sir, this is amazing. This is magic. Yes, this is magic. No, this is not magic. This is a formula. So here, can you see that in monohybrid cross we had 4 progeny? Yes, that means 4 zygote. In dihybrid cross we had 16 progeny. That means 16 zygote. So this is the tricks which we can use while solving the questions for the genetics. Clear? Okay. Great, sir. We love the session. Now give us some questions. Yes, I will give you some questions. Don't worry. But before that, something happened to my screen, I think so. Is it all fine? Let me check. Okay, let's give it a second to get the things normalized. So my first question will be what? It will be a very simple question. My first question will be a very simple question. And the question is, just a second, am I lagging? Yeah, now it's fine. My first question is what? That let's suppose we have capital Y, small y. We have what? Capital Y and small y. And then we have small r and small r. This is the genotype which is I'm going to give. The question is what? Yeah, now it's fine. Okay. So the question is what? Capital Y, small y, capital R, small r. I need you to calculate the, I need to calculate the ratio of heterozygous pairs. I need you to calculate the ratio of heterozygous pairs here. Okay, sir. Great. How we can do here? How we can do? See, this type of questions requires another approach. This type of questions requires another approach. Okay, great. So see, in total, how many do you have? In total, like, let's treat them as what? As the monohybrid cross only. Okay, we have a character which is what? Which is a dihybrid thing, but let's not solve it. Let's not solve it with the dihybrid thing. Let's solve it with the monohybrid thing only. So, if suppose that we were dealing with the monohybrid thing, capital T, capital T, capital T, small t, and small t, okay. If suppose that we are dealing with the capital T, capital T, capital T, small t, small t, small t, then what? Then you would have said that, this is basically what, sir? This, its ratio is 2/4. Its ratio is 2/4, okay. And then we are going to cut it, so it will be what? 1 by 2. Yes or no? Great. So what we did, we didn't consider this thing as a dihybrid thing. Okay, what we did is that we considered this as a monohybrid and this is as the another monohybrid. Okay, then we calculate the ratio of capital Y and small y, considering that it is a monohybrid thing. And again, the same thing we are going to do with the small r and small r. Same thing we are going to do with this small r and small r. So what is this? This is going to be what? This is going to be again 1/4. How? See, this was what? This was 1:2:1. So its ratio will be what? Its ratio will be 1/4. Okay. Now what you have to do? Just multiply those ratios and you're going to get what? You are going to get the answer which is a what? One second. 1/8, which is 1/8. Clear? Clear to everyone? This is what this will be, 1/8. I hope so that this is clear to each and every one of you. Clear? Now what, sir? Now what you have to do? Out of 16, how many things? Out of 16, not calculating about the out of 16, right? So 1/8 of 16, what it will be? It will be 8 divided by 16, and this is how you are going to get your phenotypic ratio. I hope that this thing is clear to everyone. Yes or no? Let me know in the comment box. Let me know in the comment box. Okay, great. Now, great. How we got 2/16? Are you asking this? So see, just multiply it now. We have the 16, so 1 into 2 and 8 into 2 will be 2/16. Clear to everyone? Is it clear? Great. Now let us move on with some PYQ questions. Let us move on with some PYQs questions. Great. Yes. So let's talk about the PYQs. And do you know that the PYQs questions are very, very easy? They are very easy, seriously. The first question I'm going to give you, you will be like, this type of question is asked, and this question is from NEET 2023. Okay, so the question says that a heterozygous pea plant, okay, the first thing which we got from the question is that they are heterozygous pea plant. Okay, sir, heterozygous pea plant. How many types of flowers they have? They have violet flowers and the white flowers. Okay, so the heterozygous pea plant with violet flowers. What type of flowers? Violet flowers. And it's crossed with what? It is crossed with the homozygous pea plant with white flowers. It is crossed with the homozygous pea plant, homozygous pea plant having white flowers. Okay, homozygous pea plant having white flowers. Then what you have to do is that they produced 40 progeny. How many progeny they produced? 40 progenies they produced. Okay, and what you have to calculate is that you have to calculate the ratio of violet and white flowers. What you have to calculate? Ratio of violet and white flowers. Violet and white flowers you have to calculate. Sir, this is a easy question? Yes, of course, it's a very easy question. If you see, if you will try to solve, this is a very easy question. Yes, how, sir? See here how it is easy. The question first says that a heterozygous pea plant for violet flowers. Okay, if it's a heterozygous pea plant, then that means this will be capital V and it will be having a small v thing. Okay, and then it says that homozygous white flowers, that means a heterozygous pea plant is what? It is crossed with the homozygous pea plant having white flowers. Okay, sir. So let's see here. Let's take it as V and V. Okay, and then they're going to make the gametes. Yes or no? Then make the gametes. So one gamete will have capital V, the other gamete will have small v, and from here we are going to get V no matter what. Okay, then what, sir? Make this thing. What we are going to get? Capital V and small v. Okay, and what we are going to get? We are going to get small v and small v. See, we have got two gametes and one was capital V small v and the other was small v small v. So this is going to give us what? This is going to give us the violet color, yes or no? And this is going to give us the white color. So the phenotype is what? 50-50. That means 20 among 40, 20 will have violet flowers and 20 will have the white flowers. Clear to everyone? Yes, if it's clear, that's very good. So this type of easy peasy questions comes from the Mendelian dihybrid and the monohybrid cross. So you don't have to worry about these types of questions. It's very easy. Okay, great. Now let us talk about some of the special crosses so that we can finish it off this section, which is Mendelian genetics, and we can start the post Mendelian genetics. Okay, so we have some special crosses here. We have some special cross. We have some special cross here. What is special cross? See, number one, we are going to talk about the back cross, and in that back cross we are going to talk about the reciprocal cross and test cross. Okay, so what is back cross? First of all, let us discuss that. We have back cross, and back cross is of two types. Sorry, we have out cross, and we have test cross. Okay, we have out cross and test cross. So back cross basically refers to what? Cross with the, I'm defining the back cross. What is back cross? Back cross is basically cross between parents. Okay, suppose that you had a like this was capital T and this was small t. They produced a baby or progeny which was capital T and small t. Okay, now if we are doing the reverse reproduction, that means crossing the progeny with the F1 generation, it will be what? It will be known as back cross. And suppose that if we are doing what? If we are doing crossing the F1 generation with the recessive one, then it will be known as what? It will be known as test cross. And if we are doing with the dominant one, it will be known as out cross. Clear? This is what we have. So out cross is basically what? Out cross is basically crossing of F1 individual with the dominant parent. Test cross is what? Crossing the F1 individual with the recessive parent. Is it clear to everyone? This is what do we need by the back cross, which is of two types, out cross and the test cross. Clear to everyone? Just remember that in test cross, the ratio is basically what? It is always 1:1. It is always 1:1. Clear? That's great, sir. Now, how we can do the back cross? Let's see an example. Let's again have tall one, which is capital T, short one, small t, small t. Okay. We are going to have the progeny of F1 generation, which is capital T and small t. Then what we are going to do? We are going to back cross it, which is test cross. Okay, if we are using the recessive one, then it will be test cross. If we are using the dominant one, it will be back cross. Okay, so here it will be out cross. So here we are using the recessive one, so it will be known as what? It will be known as test cross, since we are using the recessive one. So recessive one is known as what? The recessive one is known as the test cross. Yes or no? Can you find out the cross here? Can you figure it out what kind of genotypes we are going to get? Yes or no? Okay, I'll do it for you. Don't worry. See, so here in this thing we are going to get two types of gametes, sir. Yes, very good. Two types of gamete we are going to get. And in this case, in this capital small t is going to give us just a single type of gamete, which is small t. Okay, now what? Let's do mix and match. This we have this and this we have this. Sir, can we do this same thing with the Punnett square? Yes, definitely. How to make a Punnett square for this case? See here. We have, what is this? Wait, I'll draw a very good square. Yeah, got it. So we have what? We have two types of gametes in one case, which is capital T and small t. See, I am writing here this capital T and small t. Clear? Okay, sir. Now, this parent is going to give you only one kind of gamete. So let's write here small t only. Then do the mix match. Capital T, small t is going to give capital T and small t. Yes. And this small t and small t is going to give you this. Clear to everyone? So this is what? This is the test cross. And can you see the ratio? Like total number of progeny is what? 2. And one is the dominant and the one is the recessive. So the ratio will be what? It will always be 1:1. It will be always be 1:1. Tall and dwarf 1:1. And what about the genotype also? The genotype also will be 1:1. See, one genotype is like this, one genotype is like this. So 1:1 genotype also. So this is what? This is the test cross. Okay, clear? Now I'll make you revise all the things whatever we have discussed in the Medelian genetics. Okay, so first what we discussed the monohybrid cross. Yes or no? Yes, sir. In the monohybrid cross we discussed what? We discussed about the F1 and F2 generation. In F1 generation we had what? We had all tall plants. Yes or no? And all those tall plants were having what? Were having capital T and small t genotypes. That's great, sir. Now if I talk about the F2 generation, so in F2 generation we had a phenotypic ratio which was 3:1, and here the genotypic ratio was capital T 2 capital T small t and 1 small t small t. That means 1:1:1:2:1 ratio was present in monohybrid cross of the F2 generation. That's great, sir. That's very good. Now what we have discussed? We discussed about the dihybrid cross, right? Dihybrid cross. And dihybrid cross F1 generation, what we had? All dominant. What we had? All dominant. Yes or no? Yes, sir. What was the genotype there? The genotype was capital Y, capital R, small r. Right? Okay, great. Now let's go on with the F2 generation. In F2 generation, sir, what happened? We had 9:3:3:1 ratio for the phenotype. Yes or no? And you might be confused that, sir, how can I learn what is the genotypic ratio of this particular thing in dihybrid cross? My single answer will be, it's not required. Just learn the phenotype. It will help you to solve all the questions. Okay. Now, next what we discussed tips and tricks to solve certain questions which are asking about like the number of progenies, number of zygotes, number of genotypes, types of phenotypes. Okay. So for the phenotypes, what we discussed, phenotypes, we had a trick, right? So if you are standing in front of a plant, you will see it. You will see the phenotype. So let's give it a lowest number. So lowest number on our list will be 2. So 2 upon 2 ka power n, 2's to power n. Okay, the outer one. Then we had what? If someone asks you the number of genotypes, how many genotypes are produced? If someone asks, enter inside it, into the cell. Okay, we are entering. So plus 1, 3n. Okay, if someone asks you that number of zygote or number of number of offsprings, so sir, what we can do? We can do that 4:power n. So this is how you're going to find the ratios when they are asked in the exams. You're going to find the numbers in this way. I hope that this thing is clear to each and every one of you. Now here I have a very special announcement. Tomorrow I will be having a great session at 11 a.m. Tomorrow is Sunday, and on Sunday we will be solving some quiz. So since our genetics is not finished, but I have taken one short session on cell cycle. So tomorrow 11 a.m. I will be picking those questions which are very hard and have high priority in NEET 2024. So if you want to excel the cell cycle, join tomorrow at 11 a.m. and don't be late. So till then, bye-bye everyone and subscribe to this channel.